So like we said here, the final observations that you are going to make, you are going to obviously pull the leaf out, take the pictures of it, pull it out, maybe take pictures of the part you couldn't see before, okay? Then take that little bit of rubber glove off, pour the remaining liquid from the graduated cylinder into a large enough graduated cylinder to actually measure its volume, what's left in there. Then you subtract your final volume from your initial volume, and you'll have the amount that was used by the plant. Everybody follow there? Okay, so we'll have our pictures taken and our volumes recorded. Then we just rinse out the graduated cylinders, okay, and put them back in the cupboards, throw the plants in the garbage, and clean up the lab stations. All right, it's just going to go really, really quick because we don't have to do very much over there. All right, so things to watch for, okay, obviously, okay, in this one here, these were my before pictures, okay, in my after pictures, it's not really showing up very well on there, but there's not a whole lot of difference, but there is a bit of a red tinge kind of to everything. On the one I used here in the window, okay. Unfortunately, the time-lapse video, the leaf's not very well focused. You can only see a little bit of stuff, but I am going to show you something that the camera did catch that is really cool and important that we talked about in this unit that was completely by accident. Really the only things that I noticed in the time-lapse video for this was there was a bug that was crawling on it for a day and a half. And also that there is a little tiny bit of blue, okay, that's crawled all the way up to here. So it did definitely transport water all the way into the leaf. Okay, I can see evidence of that in basically all parts of the leaf. One of the best places to look is actually on the edge of the leaf. When you're comparing, the veins on the edge of the leaf may show it a little bit better. So when you're looking at your leaf, that's a place to look, right? And look for any places, obviously, where you can see the blue dye. Make sure you get good pictures of those. You're going to have a lot of the blue dye near the bottom, obviously, because it could move up there even by capillary action as opposed to like actually through the plant. So that some of that stuff on the bottom may have actually been that it just from the outside. It's not really on the inside. Okay, but you can see obviously that a lot of the dye ended up in there by looking at the bottom of it. Okay, so dye definitely got in. And then, like I say, you just got to do your measurement. Okay, my dyed water was almost right to the top, and now it's actually below the level of the glove now. So it did decrease. Okay, like I said, it was 2.5 mils. Okay, is how much I lost in there. All right, questions on what you're doing when you get across the hall there? Okay, Jake, question? Yes, this is your observation. So that's a good question. In your lab report, I'm going to want to see your photos. So you could say day 0, day 1, day 2. Okay, for your photographs, and then obviously your initial and final volumes. Okay, I mean, really, this is what your observation should look like, but in the observations section of the lab report. Okay, that's really all the data that you are collecting. There's a few pictures, as well as the final volume there. All right, so let's head over to the Chem Lab and get our plants all taken care of there. Oh, I guess I better keep that and show my other classes. Okay, so just make sure you email them to your group so that everyone can put them in their Google Drive and insert them into their lab report. Now, I am going to show you the video that we took here over the course of those few days. Unfortunately, the leaf's not very focused, so you can't see much about the leaf. However, it does show an excellent demonstration of the effects of turgor pressure, all right? Because my plant that's sitting over there that was right beside the leaf had not been watered in some time. And I watered it because I put the leaf beside it and noticed it looked like it needed water. Well, you can actually watch it in the video time-lapse recovering its turgor pressure and going from being all limp and kind of downward to being back upwards, okay, and having the leaves all be kind of, you know, upright and firm again. All right, so you can kind of see that as well. Okay, so just see how, well, that'll move back here in a second. That's when I water it. Okay, but you don't see much in the leaf. You can see a little bit of blue starting to appear. Here's the bug that's running around. He's on there for quite a while, actually. But you can watch here. See the leaves? Okay, so it's recovering its turgor pressure, and those vacuoles are filling back up, and the plant is becoming healthier again. Okay. And you can see them over here as well. So the video is not a total loss. Okay, but you can see in here as well that it's starting to get a blue tinge to it. Okay, which you can see a little bit in that leaf. Okay, but other than that, you don't see too much of the movement of the water into the leaf. Okay, but you can see that the plant is fully recovered there on the right hand side. I did, but I told you that. I did. That just shows how good you listen. All right, so again, you can see a little bit of the blue starting to show up here in the upper left of the leaf. Okay, that's that one spot that kind of showed that the water had gotten in there. But unfortunately, that didn't show. The rest of it didn't show very well. But you could see very clearly that the plant recovered its trigger pressure. So, like I say, the video wasn't a total loss. And then that's it. So, yeah, that's neat. Okay, so remember that's due November the 8th. Okay, that lab report.